previous one was the BGG net or the sequential net. Now it is attention guided. So I think for the first semester students may be helpful in the machine learning uh, for this kind of hands-on or some practical example. So today with me, with this session, assisted or assistantship by Joydeep Roy, our one of the research scholar of our lab. So I think you will do some um, that is RaceNet, I think. Joydeep, RaceNet, na? So
Yes, sir.
yes, sir. So I think you can skip some of the same introductory parts. I think everyone know as you as the total thing is I think they have understood. So I think you should go the precise and those which parts are most important for the implementation of anything of this cell biology. So okay, and this kind of thing, how to uh, go your main curriculum on the main hands-on. So you can start, Jyoti. Yes, yes.
Yes, Jyoti.
Thank you, Mehul sir, for giving me this opportunity to present today's tutorial hands-on practice using the microscopic image to classify normal or abnormal precancerous cell with attention-guided combination neural network. So, good evening, everyone. So, as my outlines were like introduction to microscopic images, importance of microscopic image collection, types of microscopic image collection, data set overview, then methods of microscopic image classification, BG16. Why should you use BG16 and why should also be used ResNet architecture and the implementation. As our sir told that these parts were previously said or explained, so I'm telling very shortly about these things. So what is microscopic images? Like it is an image enable visualization of biological cell and tissues using different types of microscopes. In cancer research, microscope imaging is extremely important because it helps us identify early changes in the shape and the structure of the cell. So, some skipping some slides. Let me tell about the dataset overview. I will be using AGMC TU paper smear cytological image dataset, which is developed in AGMC Tripura University for automated cervical cancer detection through microscopic paper smear images. And it is considered to be as a binary class. Which includes 100 images, 50 normal and 50 abnormal, which makes it a balanced binary classification data set. Each image also comes with a pixel level marks, helping the model learn exactly where abnormal nucleus regions are located. Since the data set comes from the Tripura, it also takes the overview of our region, the northeast region, that how it works in our area. Overall, the dataset serves as a foundation for building and testing deep learning-based systems like BG16 or ResNet. Next, my dataset will be HARLEB dataset, which is also a publicly available dataset categorized by seven types of seven types of different class. It is a multi-class dataset containing 917 cervical cell images. This class includes 3 normal types and 4 abnormal types. Three normal types are like superficial, intermediate, and columnar cell, and 4 abnormal types representing different stages of dysplasia and carcinoma. Because it has more than 2 categories, we use the dataset for multi-class classification, which allows the model to not only detect abnormal cells but also identify the specific stage of abnormality. All images in the dataset were labeled and verified by expert cytologists, ensuring high quality ground truth. Moving towards the next slide, uh, what are the types of microscopic image collection? As earlier we used to see that traditional methods were there for classification of the cells, microscopic cells images. Traditionally, researchers used machine learning techniques. In these methods, we first extract features manually, such as textures using LBP or HOG, means histogram-oriented of gradients, shape descriptors like the nuclear size or color features from the stained image. These features are then fed into classifiers like SVM or random forest to predict whether uh, the cell is normal or cancerous. However, the problem with the traditional method is that they depend heavily on handcrafted features. The accuracy depends on how well we choose these features, and they don't generalize well when the dataset changes. That's where deep learning comes in, like CNN and the other neural networks, which automatically learns useful features from the image themselves directly from the pixel data. Architecture like BG16, ResNet, and ResNet have been very successful in biomedical image analysis. The biggest advantage of deep learning is that it provides end-to-end learning, meaning the system learns to extract features and classify images simultaneously without manual intervention. This results in higher accuracy and better generation, especially for complex medical images. So there is a difference why we should use deep learning over traditional methods. So why we are focusing on deep learning methods like BG16 and ResNet instead of traditional machine learning methods? The main reason is feature learning. Traditional methods requires us to manually extract features from images like texture, color, or shape. This process is not only time consuming but it may also miss subtle patterns that are important for detecting cancer. In contrast, deep learning models, specifically CNNs, learns features automatically from the raw images. They can capture complex patterns, spatial relationships, and subtle changes in the cell structure, which are often invisible to handcrafted features. Moreover, CNNs provide end-to-end training, meaning the model itself learns both the features and the classifiers in the single pipeline. This reduces human bias and increases robustness to variations in staining, image quality, or noise. Finally, deep learning models scale well with large dataset, even small dataset also. For example, the HALEB or the AGMC2 dataset. Here is a short difference of BGG16 versus ResNet. As my earlier mate has explained about the BG16, it is a sequential CNN architecture which carries 16 layers. It stacks 3 into 3 convolution layers to learn features hierarchically. While it works reasonably well, it has a huge number of parameters, around 138 million, and can overfit or smaller complex dataset. ResNet, on the other hand, introduced residual learning using skip connection, which will be discussed in the further slides. These connections allowed the network to bypass layers and help solve the vanishing gradient problem, which is also be discussed in the next slide, why vanishing gradient is a problem in the deep neural network, enabling much deeper networks to train effectively. Despite having 50 layers, ResNet 50 is a more parameter efficient than BGG16 and achieves better generalization on complex or multi-class dataset. In short, BGG16 is simpler and good for small dataset, but ResNet is perfect for medical image analysis. So, what are the challenges with deep neural network? So, let's see why we use ResNet when we train a deep neural network. We update. Actually, these are the problems faced during a deep neural network. In the uh, while a input image has been trained in the artificial neural network or deep neural network, there it happens back propagation. but deep network face vanishing gradient issues while using the chain rule during the backpropagation, because when the input image is shared over this layer, that in the hidden layer, and then the output comes and does not match with the predicted output, then hence it goes back to the hidden layer. While going back to the hidden layer, it faces the gradient issues, because at that time uh, chain rule is observed. So, as we go backward through the layers, each step involves a multiplication of small derivative values with multiple layers. These products become extremely small when you reach in the suppose there are 3, 4 to 5 steps over this layer, and up to coming to this layer, the value becomes very small, and when they reach the earlier layers, the gradient almost vanishes. So this is a effective problem in the deep neural network. Earlier layer stops learning effectively, leads to slow or failed training in deep models. In that case, ResNet helps to fix this shortcut connection. So this is called the vanishing gradient problem. The network stops learning properly because the early layers can't get enough signal to update their weights. To overcome this, ResNet helps because it introduces skip or residual connection, which allows the gradient to bypass some layers and flow directly backward. The simple idea ensures the model can be trained much deeper, like ResNet 50 or ResNet 101. Let me explain about this. This is a normal network layer which a input image is converted to is going to a convolutional layer, and then performing the batch normalization, the pooling, and the ReLU and the pooling layer, then again to the convolutional layer. It is a normal network of deep neural network. But what happens in the ResNet? It follows uh, it follows the network like a residual mapping. From X, if we take X as the input, and then it comes to the convolution layer, performs batch normalization, ReLU, pooling, again to the convolution layer. But in this case, they are happening three direct layers. But while using ResNet, it skips some layers and directly goes to the output layer. We'll consider FX as the output layer. So the final output becomes hx, means the x equals to fx plus x, because the residual mapping is fx equals to hx minus x. The learning should be like this, that the x means which carries some information is directly going to that addition symbol that carries information and resolves the gradient problem. This shortcut allows gradient to flow directly to the earlier layers during backpropagation. This prevents vanishing gradients, enabling the training of the deep neural networks. These clip connections means the gradient does not have to flow through every intermediate layer. It can directly reach layers, so even if some gradient becomes small, the shortcut helps to preserve them. So it is the residual network is denoted as hx equals to fx plus x. Moving towards the next slide about the ResNet architecture. If we consider an input image of 224 into 224 of 3 channels and into 3 channels, in the first layer of ResNet, the input image passes through 7 into 7 convolution layer with 64 filters and a stride of 2 and a padding of 3. The ResNet 50 the structure is like this, that it first the initial layers comes with the 7 into 7 convolution filter, then max pool, then 1 into 1 convolution layer and 3 into 3 convolution layer and 1 into 1 convolution layer with 256 filters will be continued 3 times, that is 9 times. Then again with the this into 12 times, and again the same type of convolution layer of 4 will be converted into 6 times, so it comes into 18, and then again in 3 times, which comes in 3 into 3, that means 9, and at last there will be a average pooling layer with fully connected with 100 neurons. This actually, this is the actually the 50 layers of ResNet. Now let me explain how it uh, solves the each layer, because in the earlier slides or earlier session you have seen how convolution works. Now I'll be showing that how ResNet works in this convolution with this formula, that n plus 2p minus f by s plus 1. There is nothing, just normal convolutions. See here, if you take a input image, here converts it to the initial layer, that is 7 into 7 convolution with 64 filters and the stride of 2. Here is the first layer explanation. See, the input size n is 24 to 24. The filter size is 7 into 7, which type 2, the padding is 3, and the feature map or the number of filter is 64. We are not explaining going to the number of filters, because the about the filter thing in the earlier session it has been explained. Output size equals to how much will be the output size, depending on this formula. In input size plus 2 into padding minus filter size divided by the stride plus 1, it will decrease the uh, output size will be decreased in every each layer. See, when it comes 1125 into 112.5, since we take the convolution layers as floor values, the output size will be 112 into 112 into 64. This is just an example. The input size will be can be anything. It can be 116. Here we have taken the input size as 224 into 224 into 3 channels RGB. In the next, sorry, here is some problem. Okay, in the next